Yeah, I have to swap this.
Hey, hey. Test, test.
It's being streamed.
Test one, two. Hey, hey. Test one, two.
So it's on.
Okay.
If you're gonna say anything, actually, you know what? If you're gonna be here, can you put it in the middle? Because your head's gonna be going this way towards the screen.
How's that? Good. I talk really loud.
Perfect. And this can go in an inside pocket or outside coat pocket.
Okay.
Whichever.
Thank you.
See if someone's gonna do an intro.
Oh, hey.
Thanks for coming.
Thanks for inviting me.
Yeah. We usually wait. They're in a different location than we usually are because
Yeah. It's a nice room.
hope it works out for you.
No, I'm sure it'll be great. Hey, how are you now? Good to see you. Thanks for inviting me. Good. So he was explaining to me we usually wait a little bit.
Well, especially today because we don't usually have it in this room.
Do you think you want the lights down, or do you have any slides?
Not really. No, doesn't matter. Whatever you guys want to do. Harvard's super nice. All the buildings and everything is very pretty. Fancy.
Present to you Jeff Schonbaum, who's visiting us from NIH. And rather than boring you with tedious academic lineage, maybe I'll just editorialize and explain to you why Jeff is one of my scientific heroes. So as probably all of you know, there's been this long history of interchange between animal learning theory and experimental neuroscience. The problem is that a lot of the learning theory that has been employed by neuroscience is basically 30 or 40 years old and more or less wrong in some fundamental ways. So we're in this kind of funny situation where we have this fantastic neurobiological toolkit. We're applying it to testing theories that are basically wrong. And so enter Jeff, who is open-minded and adventurous enough to try to embrace sort of the new, the brave new world of computational learning theory, and really try to both apply that to designing new experiments and also to try to challenge that learning theory, challenge our assumptions about it, and push theorists to develop more empirically accurate understanding of how animals learn. So just as an example of that, a few years ago, he, in collaboration with Bob Wilson and Yale Niv, pursued this idea that the orbitofrontal cortex was important for representing state spaces. And I think that the whole notion that a state space representation, the very notion of a state space at all, I think would be very alien to a lot of neuroscientists, but now it's been at least partially absorbed into our thinking about the OFC function. So that's just one example of the way in which Jeff has kind of assimilated the current thinking in learning theory, but also he's challenged it, and I think that's really where I've derived a lot of pleasure from reading Jeff's work, which is that he finds all these surprising things that I didn't see, and it forces me to rethink and question my assumptions. And so I'm really looking forward to hearing what Jeff has to say today and the new challenges that will present us with.
Great. Thank you very much for the introduction, Sam. So if you're surprised, imagine how we feel about it, right? Okay, so thanks for inviting me and for coming out to see my talk today. So I'm going to talk about two experiments we've done to test what seem to me to be really critical predictions of the idea that dopamine transients are functioning as cache value prediction errors in the brain, or at least that is really the full description of their role and what they can do. So I'm just going to talk basically about two experiments. I can definitely take the whole hour, but I can also compress it. So if I'm not making sense and you have questions, I would invite you to go ahead and interrupt me, and I'll try to explain better. I think that usually works better than holding all your questions until the end. And if I need to, I think I can compress the end of the talk so we can all get out of here on time. Okay, so that as an introduction, I'll go ahead and get started. just to make sure we're all on the same page, I wanted to start with this iconic figure here that I imagine everyone here has seen 100 times before, just to illustrate exactly what I'm talking about when I talk about dopamine transients and their resemblance to prediction errors. And so this is a recording made from a single putative dopamine neuron from monkey substantia nigra in Wolfram Schultz's lab. And what you see is that this neuron has a nice phasic response lasting about a half a second or a bit more when a reward is delivered. But this response is not simply representing the occurrence of the rewarding event itself, because down below you see that if that reward is preceded by a conditioned stimulus, a cue that predicts the occurrence of that reward, the activity of neurons like this decline, and in this particular spectacular case, go completely away. And the loss of this activity or its diminution is not just habituation or the animal not paying attention anymore. We know this because at the bottom you see if you deliver the cue but omit the reward, first of all, the monkey definitely notices, right? But more importantly, the neuron notices as well, now suppressing its firing for about the same amount of time, again around the time the reward should have happened. And so this fingerprint of activity is providing a really nice correlate or facsimile of a reward prediction error, right? So it's increased when the reward is better than expected, reflecting a positive error. It declines and can go away when the reward is completely as expected. And then it's suppressed or goes down when reward is worse than expected, reflecting the negative prediction error. And so that's really the kind of dopamine transient that I'm talking about here. So this correspondence was recognized about 20 years ago, I think by a number of people, most prominently Wolfram Schultz, Peter Diane, and Reed Montague. And what they said at the time, and I think have maintained since, is that this pattern of firing and other features it shows, I think most prominently the transfer of the response to the antecedent cue to the conditioned stimulus is not just any old prediction error, and it's not a reward prediction error that you might have seen in lots of other theories, but is specifically the prediction error contained in so-called model-free reinforcement learning algorithms like this one from Sutton and Bartow. And forgive me, I understand there are probably lots of these. This is the one that's always mapped onto the firing of the neurons, and so it's the one I show. So what these guys said, as I understand it, is that the change in value that happens in a learning situation like the one you saw in the prior slide is going to be equal to this constant that reflects the salience, sociability, or learning rate supported by the materials at hand, multiplied by this error term that they called delta t. And so delta t is just the difference in the value of what's actually experienced, so both the intrinsic value as well as the value it has from what it predicts about the future, right? So that's why the transfer back to the cue was so influential in their thinking. So the sum of that value minus the value that was expected going into that time slice or state in the model. And so the strong version of this hypothesis is that the transient firing of the dopamine neurons, like I showed you in the prior slide, is this delta t, right? That dopamine is the biological instantiation of this teaching signal. So, so that's the idea. And to be honest, when we first started recording dopamine neurons in the lab about 10 years ago, I actually didn't focus very much on the implications of this specific idea. I thought it was enough that these things were signaling what looked like prediction errors. I mean, the neural correlates are really amazing for their specificity. But more and more, I think the details of this hypothesis are really important. And the reason I think they're important is because delta t lives within a really simple, and I would argue quite impoverished learning model, right? And I say that because delta t basically only knows about the value of what just happened in these models, right? So something was either better than expected or worse than expected, right? And it reflects that. But it doesn't reflect anything about why it was better or worse than expected, right? So if you have a terrific meal, that induces a positive value error. If you win the lottery, that induces a positive value error. This signal might differ in magnitude, right? But it's not going to help you keep track of those differences in any other way. Not only that, it also is not going to reflect anything that changes that doesn't impact value, right? And of course, we all know, we learn when things change in the environment that might be orthogonal to value. Formally, It only acts to increment or decrement the value that's cached in the antecedent events, right? So it increases or decreases how much we value them, I guess. But it doesn't actually drive real associations to form between those events and the outcomes that are following them to change that value, right? And it's a closed loop system, so that this value that's cached in the events feeds back and becomes what they predict when they're encountered the next time in order to calculate delta t again, the next step. And so it seems to me, while this might be a terrific way to train agents to wander around virtual mazes, it really does not capture the way, first of all, that we want to be taught about things, what we hope that we're learning, right? This sort of warmer, colder way of teaching. And it definitely doesn't capture what we want to learn about the world around us, at least on our best days, right? In addition to this kind of cache value learning, we certainly want to be learning about the causal structure of the world we're moving through, right? So that we can have a mental map or a cognitive map of that structure that we can navigate virtually in order to make good decisions in the future. At least that's sort of how we hope to operate on our best days. So that's what troubles me about this. And just to contrast this for you, I think this is really different than the concept of prediction errors that comes out of experimental psychology. And it's exemplified by Rescorla Wagner, but I think it's shared by most of the, certainly the older learning theory models and really the newer ones too, right? So here you have a simple model that has, for my money, most of the same parts and pieces you see in the other one. I think that's no accident. I think they were inspired by this. But notice what these guys said, whether intentionally or by accident, is that these variables operate not to increment or decrement some general value, but to change the strength of associations. And here we're talking about the brain, right? So I think it's a reasonable extrapolation to say that you're talking about associations between the neural representations or the neural pools of these external events, right? So you put an animal in a box, you turn on a tone, you deliver a reward, right? The neural representations of that tone and that reward become linked, become associated. Now you might conceive of the value as being those synaptic connections, the efficacy of those connections, right? And that's fine. But fundamentally, when the animal encounters that cue again, it's not going to evoke a representation of the value. It's going to evoke a representation of the food, right? Of the actual physical thing that was delivered. So that's the kind of real-world associative learning that errors were originally developed to explain, right? And that I think was captured in these models for me, at least when I was a graduate student. So this is kind of the contrast that drove the work I'm going to tell you about. So to go back to our biological signal of dopamine, right? Dopamine shows this error signal. And so I think it becomes really important to know, is this signal fully described by this simple computational model, right? In which case, great, we need to be looking for some other signals. Or might it do something more than this? Could it have a broader role than is envisioned by this model? So that's the idea. Now, of course, you may be thinking to yourself, we now have lots of evidence that irregardless of my introduction, the dopamine signal does in fact behave exactly like we would expect for a cache value error signal. And that's in fact true. And this is just a small fraction of these studies. These are some of the nicest studies that have been published during my 20-year career. They're really the most amazing research. The data is beautiful. And the finding is easily replicated across labs, across learning materials, across tasks, across species. In the right circumstances, dopamine neurons behave as if they are a cache value error signal. But I think there's two things you need to keep in mind about this impressive array of data. The first is that these are almost entirely correlative studies, right? So people change the value of what the animal expects, and they show that it influences the spiking activity of dopamine neurons, dopamine release in accumbens, or in some cases, bold response in places that get a lot of dopamine innervation. And I would suggest that while that might tell you what information is in the signal, it doesn't tell you what kind of learning the signal supports, right? The second thing that strikes me when I think about these data now is that by and large, and I really think entirely, they confound changes in cached value with changes in all sorts of other features of the reward itself. And you don't have to think hard about this to realize it's true. Just think about the example I started with, right? So in that figure, they're inducing a positive prediction error by delivering a reward the monkey doesn't expect to receive. So sure, maybe you've changed the value, you've delivered something that has a value that wasn't expected, but obviously you're changing all sorts of other things, right? So the reward has a timing, a taste, a texture, a temperature, right? All these features are also changing and are unexpected. They might relate to value, but it's not the same as just changing value. The same is true when you omit a reward to induce a negative prediction error, right? There you're leaving out an entire sensory event that the animal has been anticipating. And I think this is true whether you induce errors, value errors, by changing the timing of a reward, changing its magnitude, changing its number, changing its probability, or even if you switch between two different rewards that have different values. In all cases, you're confounding cache value, which is what you're trying to isolate experimentally, with changes in other things. And I think that is just fundamentally not good experimental practice. If you think something signals cache value, then it's incumbent on you to isolate it. And that criticism, by the way, applies not only to these correlative studies, but also to the growing number of causal experiments in which people are using optogenetics to manipulate the firing of these dopamine neurons in temporally specific ways in order to ask, will they serve as teaching signals, right? And these are just two examples, one from Trisha Janik's lab, one from our lab. In each case, both experiments have tried to experimentally eliminate just the prediction error and leave everything else the same and then put it back by stimulating these neurons or inhibiting them. And in each case, their study and ours, we're able to recover what looks like normal learning, right? But in neither study can you distinguish whether that learning is driven just by cached value, right? Or does it reflect this sort of richer associative structure? So that's the goal of the two experiments I wanted to talk about today, was really to put at risk the key predictions that dopamine neurons act only to drive cache value learning, and that they only reflect information about changes in cache value. Okay, so I'm going to start with the first one, which is going to be to look at the second part of that question. So basically, the aim of the study I'm going to talk about first is to test whether the content of the learning supported by a dopamine transient is limited to this general or cache value, or whether dopamine transients are able to drive richer associative learning, the ability to link a cue to predict something about what is actually coming. So there are a couple ways we could have done this experiment. And we chose to use something called sensory preconditioning, and hopefully as I describe it, it'll become clear why we made that choice. It might seem overly complicated at first, but we did it for two reasons. The first is that we wanted to test this hypothesis in a setting that was divorced from the delivery of any sort of a valuable reward, right? And that's why the sensory preconditioning is going to be important. So we're not going to confound whether dopamine transients can drive learning with presentation of something valuable, right? The other reason that we're going to use it is because, as you'll see, it allows us to ask whether dopamine transients can drive associative learning in a setting where the ultimate response we're going to use to assess whether the learning has happened is not present, right? So that gets us away from a confound where the dopamine transients might be just stamping in that response. All right, so what is sensory preconditioning? So sensory preconditioning refers to a simple associative task developed by Brogdon, actually working at Johns Hopkins in the 1930s. So it's a way of showing that animals form associations between neutral cues. And so what you do is you start out in the first phase by presenting the animals with pairings of two neutral cues, say a 10-second tone followed by a 10-second white noise, A and X. If you do this a bunch of times, it turns out the animals actually will learn that A predicts X, right? There's no evidence of that here because you're not asking the animals to do anything. They're just sitting quietly in a cage. And so Brogdon came up with the idea of revealing that learning by taking one of those cues, usually the second one, and pairing it with something the animal wants, an unconditioned stimulus. And so for our hungry rats, that'll be food pellets. And so we take X and pair it with delivery of some food pellets. And of course, what the rats do is they develop a conditioned response during X of going over and looking for food. Right. Once that happens, we then come back in a probe test and we present A and X alone, interleaved without reward. And we ask, what does the animal do? And if we do this right, what we find is the animals will now show a conditioned response not only during presentation of X, right, the 10-second cue that predicts food, but also during presentation of A, the cue that predicts the cue that predicts the food. And they show this response to A despite the fact that we've never presented A again, A paired with food, and we've never presented A again with X after X has been paired with food. Right? So the only logical way to explain why they're looking for food in the final test when we present A is if A has acquired the ability to evoke a representation of X, right, in the first phase. And then, of course, when X predicts food, they make the inference that if A means X and X means food, that maybe food is coming. Right? In fact, you can show that experimentally before the probe test if you devalue the food by pairing it with illness or satiating the animal on it. What happens is the animals will respond less, the rats will respond less for food, not only during X, the cue that predicts the food, but also during A, right? So they really are responding to A as if they think now food is coming. So our idea was that this provides us with an excellent vehicle to test whether triggering the dopamine neurons to fire. In a way, we know they do when the animal encounters a sensory event that has value, so an unexpected food pellet, for example, whether driving those neurons to fire here at the start of X would actually be able to recover real associative learning, right? Associate A and X, as opposed to this sort of cached value representation. So our idea was that we would essentially block learning about X, the same way you would block learning about US by putting another cue there that already predicts X, decrease the informational error that must be driving the learning, and then see if we can put it back by triggering the dopamine neurons. So our first step was to go look in the literature for a study that we could copy basically to block sensory preconditioning. To the best of my ability, I've been unable to find anyone that has done this. So if anyone knows of a paper from 40 or 50 years ago showing this, please let me know. We weren't able to find one, and that's actually important because it might be that just putting two sensory cues next to each other is enough to cause them to become associated, right? If there's not some sort of an informational error driving learning, then putting the dopamine signal in wouldn't make as much sense. And so the first thing we did was we developed a paradigm to test basically whether this was an error-based form of learning. So for that, we did exactly what I showed you in the previous slide. We trained rats that A predicts X, and then we're going to come back and pair X with food. And in between, we inserted this blocking phase. So this is exactly what you would do if you wanted to block learning for a US, except instead of a US, we're now using X as the second cue, right? So the blocking phase has 4 trial types. The bottom trial is just a reminder trial. So it's just like the trial type here, so you can sort of forget about that one. The next 2 trials are our critical blocking trials. We have 2 of them because later I'm going to need to. In the pilot study, they're exactly the same, so you can think of them as the same. So here we're presenting A, this 10-second cue that already predicts X, and then we're pairing it up with another auditory cue, either C or D. These are just clickers, buzzers, tones, other cues that we have available, followed by X. And the idea here is that if learning to predict X is driven by some sort of an informational prediction error, think of it as a sensory prediction error, then because A already predicts X, that error should be diminished, less. As a result, the animals should not learn as well to use C and D, or to associate C and D with X. And this will be evident, of course, not here, but after conditioning in our probe test, where we're going to present C and D, and our expectation would be that the animals would respond less to these 2 cues than to a positive control cue, F. So F is just another auditory cue that we present here, followed by X, in the absence of A. And just for good measure, because all the cues are being presented in compound, we pair F up with another novel auditory cue, E. So it's sort of you can think of it as overshadowing. So F should be weakly associated with X, but not completely blocked. So that's the idea. Here you see some of the data. So this just shows the magazine entries the animals make during presentation of each of these cues or cue combinations during the preconditioning phase. They have a little food cup shaping before we begin, but not very much. And so what we see is very low levels of responding as expected. We then go on to the conditioning where we pair X with food, and you see the animals develop a strong conditioned response across sessions. And then we do our probe test where we present these 3 cues alone without reinforcement, and we look at responding. And consistent with the idea that there's some sort of a prediction error driving the SS learning, what we see is the animals respond significantly less to C and D, our 2 block cues, than they do to F, which is our weak positive control cue. Yeah? 2 magazine entries on average in the 10-second queues. So baseline might be 1-ish. So, under 1 is sort of where we would see, and then they go up some in the really in the first session, and then they continue to increase across days. So they are showing some, I guess I would call it generalization, but clearly less to the 2 blocked queues than to the control, the positive control. So it's essentially, it's analogous to the kind of experiment that Cayman did to block US learning. And there was no reason to think it wouldn't work, but it was important to show that it does work. So this then provides us with a vehicle to ask whether putting a transient burst of dopamine firing into the equation at the start of X, right, is able to recover this kind of associative learning, right? There's no reward present. The food cup response is not present here, right? So we're not going to be somehow artificially stamping that in, right? The question is, can we recover the learning by triggering the dopamine neurons? And so to ask that, we got new rats. We used TH-Cre rats. All rats had a Cre-dependent virus infused into lateral VTA. Half of them got a virus containing channel rhodopsin, so we could activate the dopamine neurons. The other half of the rats got the same virus lacking the opsin, so these are our EYFP controls. We implanted fibers bilaterally over the area so we could deliver light to activate the opsin. And then after recovery and food deprivation, we just trained them on exactly the task I've been talking about. And to manipulate dopamine neurons, we delivered light into VTA for about a second at the start of the 10-second X queue. So the idea here is to try to cause the neurons to fire in a way that we know they would fire if the animal encountered an unexpected reward, right? And if all that does is act to cache value on the cues that are present or around at the time, right, then you could imagine we might affect all sorts of things going on here. But what we shouldn't do is selectively recover the C that we're just hijacking the normal signal that only appears when there's a cache value error and using it to drive associative learning in this setting where it's not normally involved. I still think that would go beyond what the model predicts. Yep.
So you have to continue to pair just A with actually going.
Correct.
Right?
Yeah. Well, I don't know if we have to, but we do.
Oh, you're not sure if that's necessary.
So the idea is to maintain the animal's representation that A predicts X so that there's not extinction of that, and so it basically maximizes the blocking effect.
So just imagine that that's like crucial to do for a number of trials because maybe it's supposed to depend on how long it's
Yep.
but suppose that's crucial, and then you can imagine that the ability to to block let's see would be a concern. With A depends on in some sense the ratio of the number of AD trials and AD trials to the number of A trials, right? And so if that was true, and then you gave extra dopamine, right, for association time, right? It would seem like what we could do in this sort of the reward is somehow emphasize those problems to be more important, right, more rewarding, right? Sort of effectively shift sort of like the ratio of sort of importance of how many sort of HAC files there are to pure A trials.
I guess I think.
Extreme, it would sort of act like the AC trials are so important now that it's like you don't even have any AX trials.
Right. So if we got rid of the AX trials, the prediction would be that you would. I mean, over time, what's going to happen here, especially without any AX trials, is that the learning is going to redistribute. So the blocking phenomena is something that happens initially, but if you continue to train ad nauseum, AC predicts X, AC predicts X, AC predicts X, and you didn't do anything else, right? Eventually, the learning about A would extinguish a little bit, and learning about C would increase a little bit, and the learning would distribute across A and C over time, right? And so, so that's fundamentally why we have that there. If you did the training for only a brief period of time, which actually is what we do, that redistribution would probably not have time to happen. So I think we probably don't need the AX trials. What I'm not sure I'm following is, so you're saying if we get rid of that, that somehow this wouldn't work anymore, or that the result would be invalidated, or I guess I'm not clear what I'm.
Yes, the prediction would be that sort of A, or the association of A with X, right? Say what happened in semi-training, right? Might happen, much more swiftly if you were sort of overvaluing the AC trials compared to the A trials. It means that that's overvalued.
Right. So.
So the prediction would be that if you did this in an extremely short experiment, right, then there would be little effect of what you're seeing, and as the experiment was longer and longer, the effect of the sequence more stronger and stronger.
So obviously we have to have a good blocking effect here, a big distinction. Otherwise, we won't be able to see the recovery of the learning, right? We only. This is about 12 presentations, so it's not a lot of presentations. So let me rephrase. You're suggesting we're just dropping a whole lot of value onto X, and that's causing a redistribution here that wouldn't normally happen. Is that fair?
Sure, like sort of kind of like a lot more AC trials.
That's fair. I mean, I think the difficulty is that nothing in the computational model says you should recover a real association, right? The model does not drive associative learning. The model drives value caching, right? So what that would mean is that C would be super duper valuable, right? And why would that then cause them to march over to the food cup and look for food when you present C? C is never paired with food delivery. It's never paired with X again after X means food delivery. There's no response there to become associated, right? So it sort of goes to the heart of what it means to say that you're incrementing or decrementing value.
You're not messing with dopamine at all, right? You have A trials because C will become associated with X, right? If there's a high preponderance of AC trials versus A trials, right?
So it's causing a redistribution. Yeah. So in that case, you would also expect to see changes up here, right? Because A is common. So you're arguing that the dopamine is decreasing the significance of A. So A would not block D as well either, correct?
Yes, so the prediction would be that
So I'm getting responding. Right, so I'm getting responding to C in my experimental group because dopamine is somehow causing A not to mean reward anymore, and so it's unblocking, not because it's having some principled effect on associative learning for X, but because I'm screwing everything up for whatever reason. And so A is now not predicting X very well. So now I have this control group over here where A has to predict X for D to be blocked. So if I'm messing up A, then D ought to be unblocked too, and of course it's not. D would go up. D should go up just like C does, right? Because you're arguing we're doing something during the learning phase that's causing A to change, and that's why C is becoming unblocked. A is becoming a weak predictor of X. No, I mean, I think you're making a good point that there might be something here going on that we don't know about, and you're pointing out that, hey, you have to keep A strong, and that's why we've gotten these. So maybe X is just interfering with that process and is causing A to not be a strong predictor of X, and that's why you're getting inappropriate learning to C. And what I'm pointing out to you is I have a control for that. If A is becoming a weak predictor of X, then C should be unblocked, and D should also be unblocked. You don't think that's an adequate control for that criticism?
No, I mean, if you had, say, small number of AD trials, right?
So dopamine has to be there in order for A to remain weak for C to be conditioned. So the AD trials don't control for that.
I mean, my prediction would be, would be that the blocking D might actually look stronger. It doesn't seem like the data shows that. So all of the
The blocking of D would get stronger. Huh. Okay. Well, yeah, we definitely don't see that either. Yeah. Well, I'm certainly willing to admit there might be lots of things going on here that we don't know or understand. So I don't have any problem with that. All right. But certainly within the constraints of our a priori predictions, which, by the way, I actually talked to a lot of computational and learning theory people about and didn't get any arguments about going in. I think our data definitely are showing a kind of change in behavior that shouldn't be supported if these signals are only acting to cache value and things. I mean, for one, the failure to see any change in how fast the animals learn about X later suggests that if we're changing value, it's really only happening in a transient way that goes away, right? It's not impacting how fast those things can be learned about the very next day. But it doesn't address whether any of this stuff actually normally happens in a setting where there is no value present sort of exogenously, right? So it could be that dopamine transients are driving SS learning when there's a reward present, right? So you learn the cue predicts the reward and all its sensory features, but the dopamine is not normally involved in preconditioning. And this was something that the reviewers were really interested in. To be honest, we had avoided this experiment because the only way I know of to suppress the firing of the dopamine neurons in a temporally specific way is going to be used to use Halo-rhodopsin, and that's obviously going to induce potentially a confounding negative prediction error, right, which is kind of problematic. But nevertheless, we went ahead and tried to do the experiment. And to try to avoid this confound, what we did was we suppress the firing of the dopamine neurons for a longer period of time, starting before the transition between the two cues and continuing for a couple seconds after, so about 2.5 seconds. And our idea was to try to interfere with any transient increase in firing of the neurons, or maybe a pattern discharge, some sort of an ensemble code, right, without inducing a suppression in the firing with the timing and a duration that you would normally see as a negative prediction error. And whether you buy that or not is up to you. So we again used THC rats. This time we infused a virus into VTA containing Cre-dependent virus containing halorhodopsin. The controls got the same virus without the opsin, and then we put fiber optics down so we could deliver light to inhibit the neurons. And then we trained these rats just in your basic sensory preconditioning task. So we presented them with pairings of 2 auditory cues, B and Y, and suppressed the firing of the dopamine neurons by delivering light a little bit before the end of B and then a couple seconds into Y, sort of spanning the period when we had stimulated before. Y was later paired with food pellets, and then we probed for B. As a control, each animal also learned about 2 other auditory cues, A and X. X was paired with 2 different food pellets, and then we probed for A. And the idea here is that in the control group, we should see an increase in responding to both A and B, right, because they're both conditioned with cues that later predict reward, and that increase should be similar. If something going on with the dopamine neurons is necessary for the SS learning in the first phase, then in our experimental group, we would expect to see less responding to B. So that's the idea. Again, in the preconditioning phase, we see low levels of responding to the cues. We then condition both X and Y now, so we have 2 cues we're pairing with reward. And again, we see the development of a conditioned response to both. We have a bit of an increase here that looks like it might be important, but it turns out to be there as well to a lesser extent, and there were no differences here. Both groups end up at the same level, which is important because we're going to compare their responding in the probe test. The other thing that's important is that the experimental group conditions exactly the same to X and to Y, which is the cue that we're inhibiting the dopamine neurons at the beginning of. So if you thought that was going to make Y aversive, decrease attention to it, change something about it, we don't see any evidence of that in how the animals learn about it. All right, we then do the probe test. So in our controls, as expected, we see an increase in responding to the 2 cues, and it's similar. In our experimental group, we still see an increase in responding, but clearly they're responding less to B, which is the cue we tried to prevent from becoming associated with Y, than they do to A. So I certainly see this experiment as having some significant caveats, but it's at least consistent with the idea that there's some transient change in the firing of these neurons at the start of that second cue that normally is important for the SS learning. Yeah. Now? Sorry. So I guess I would see it as more significant for this experiment. So the idea would be that because we're inhibiting the dopamine neurons, that they don't like B very much, and it causes them to freeze or do whatever, and that's why we see less responding. So I mean, I think that I don't have direct arguments against that, except for the fact that the dopamine neurons are being inhibited during presentation of Y, right? So whatever's happening to B presumably is going to be at least as bad for Y, and we don't see any changes in their learning for that.
You studied stimulation
true, but if I deliver food pellets, I'm going to get much better conditioned responding, and in fact, I'll also get much better freezing to a cue that I paired directly with concurrent shock or food pellet delivery. Doing it later only makes things worse, right? So, I mean, I get the point, but I think that the data from behavioral experiments would suggest that doing something during it is actually going to be more effective. Yeah, in any event, I see the criticism as much more significant for this. For the study in which we're stimulating the dopamine neurons and recovering a response, it's a directed response. The animals are looking for food. It goes away when you devalue, so they're not just wandering around doing stuff. They're doing something very specific, and it's a response in an activity that wasn't present when we stimulated. So it's not that they had their head in the food cup as they might have in Trisha's blocking study when you're stimulating the dopamine neurons, and that then of course you get more of that thing. It's a totally spontaneous new behavior that they engage in, right? So I don't see an easy way to argue that it's being driven by a change in value.
But during conditioning period, you are kind of training for the paradigm or setting in the paradigm.
In the first experiment.
Or in this experiment, when you are training with X and Y, you are teaching the rats what to do in that environment.
Right.
So that dopamine transient might only manifest after the conditioning phase.
Right. Well, that's why I say that I think the criticism has more applicability to this experiment, because we're causing you could be causing a general behavior to the cues that are paired with the optogenetic inhibition that affects your specific food cup response that you train later. What I'm suggesting is the first experiment, we're inducing a very specific response by the learning. It's a specific response that doesn't appear at the time, that shows up once we've given the animal the ability to express it. So I don't see, I can definitely, I mean, I can't exclude it. I mean, sure, but it doesn't strike me as an explanation anyone would have come up with in advance, right? It's not something. It's not a proposal that would explain the data.
But there must be some control.
Well, there is a control, which is that if I devalue the food, they don't go to the food cup anymore, right? So they're going to the food cup during presentation of C, not because it has some value, but because it is predicting something that predicts food delivery, right? Okay. I mean, this is the problem with the idea, frankly, is that it's almost impossible to get rid of it, right? So I guess so my plea would be, if you don't like my experiment, go come up with your own, right? Because trying to rule this out in a way that no one else can come up with a counter argument to is almost impossible. And I tell you this from experience when we did this study. This is a study that I described to lots of people in advance of doing it, and no one presented any of these objections. But since we published it, we have got a whole raft of them. Actually, the people in my lab want to write a paper about all the possible alternative explanations that people have proposed, right? And this is for an idea that we all know must be wrong, right? This is not the way that we learn, right? If all dopamine does is cache value errors, then there's a giant teaching system in the rest of the brain that we all need to be looking for, right? Because the way that we learn things is not described by by this specific computational model, right?
But no one said that all association is controlled by a single mechanism.
But nobody else has found anything in the brain that does prediction errors the way dopamine neurons do. I mean, they're amazing.
There are many prediction errors by other neuromodulators.
I don't even have to analyze the neural data. I can just walk into the room and listen to the neurons, right? I mean, they're really striking. Don't you think? I mean, you know, I can record from amygdala, and I can't pick out the neurons generally that are firing to something that means reward or punishment, right? I have to look at the rasters. The dopamine neurons, I think they're really pretty striking. So the point is, if they're going to be limited to this very simple way of learning, then okay. But it seems to me that if I have this powerful teaching system buried in the base of my brain as I'm evolving all these cortical systems, why wouldn't I try to use it? Right? So just trying to do experiments to stretch the envelope and suggest that it might be doing these other things. And I appreciate all the ways it could be still a value signal in doing this. But I really think that there are less parsimonious explanations than the one I'm just suggesting, right? Which is that we're just driving the recovery of the association the way the system's sort of supposed to work. Okay, so now I'm on to my second experiment, and I have exactly 5 minutes, and fortunately the second experiment is ridiculously easy to explain. It really goes to the heart of whether dopamine transients are simply sufficient to drive learning when there's no reward around, or might they really be signaling errors when you hold value the same and you change something else about the reward, right? I started my talk by pointing out that all of the evidence in favor of this idea comes from correlative studies mostly, where people are changing the value, and yet they're also changing all sorts of other things about the reward itself that could be contributing to or part of the error signal. And so it seemed to us that a really simple experiment that to our surprise hadn't been done was one in which you basically held value the same, right, and changed features of the outcome. And so that's the second experiment that I want to try to briefly get through. So the way we did this study was to use a simple odor discrimination task where the rat samples an odor at a port and then goes left or right to get reward. We use this task a bunch of times. The rat has 3 odors. One means go left, one means go right, and then there's a third that the animal can go in either direction to get reward. And this gives us a ready readout of which well is more valuable. Those stay the same. During recording, what we do is we manipulate what reward is coming out of the fluid wells across blocks of trials in order to induce, well, we do it for a variety of reasons. When we're recording the dopamine neurons, we do it in order to induce prediction errors. And for what I'm going to talk about, we have 2 manipulations. One is a change in reward number, which is meant to induce the classical cash value prediction errors, right? So we're going to deliver more rewards on one side the animal doesn't expect and omit rewards on the other that the animal does expect. And then in other blocks, to contrast that with that, we're going to keep the reward exactly the same and change its identity. Keep the reward number the same and change its identity. And so the blocks of trials in a session might look like this. So this would be the rewards the animal gets on one side, and these would be the rewards on the other side. And we used chocolate and vanilla flavored milk solutions that we know from past experience and testing these rats, the animals can tell apart, but they really don't have any preference between. They'll drink similar amounts of them across time if they're given both. And if we pit one against water, they show similar degrees of preference. So that's the basic idea. Here you see some of the behavior from the free choice trials. So you see when we make the vanilla reward the big reward, the animal's free choice behavior switches across 10 or 15 or 20 trials in the new block. Same if the chocolate becomes the big reward. So value or number has a big influence on their behavior. Here you see what happens to their behavior if we change the identity of the reward without shifting the number. They don't change much. They increase their variance a little bit in terms of how they're picking, but they don't show these big directional shifts. So we think that suggests they value the two the same. We also see, I think, analogous changes in behavior on the forced choice trials. So they're a little bit faster. And a little bit more accurate when the big reward is at stake versus the small. So this is big and small. And you can see the 2 flavors are exactly the same, right? So that effect doesn't matter which one is big and which one is small. All right, against that backdrop, we record from lateral VTA. And we record all the neurons, and then we use a waveform sorting analysis to identify which ones we think are dopaminergic. So we try to pick out neurons that have long polyphasic waveforms, slow firing rates, reward responsive. And the goal was really to identify dopamine neurons that would be picked out in Wolfram's lab as being dopamine neurons, right? And so that would be these guys. Neurons in this cluster are sensitive to IV infusion of apomorphine and quinprol. I think Rui Costa has shown that similar neurons have NMDA receptor-dependent bursting. We've actually gotten a fair amount of flack with this, however, over the years. So in this experiment, we included a group of TH-Cre rats in whom we infused CASP3, which is an apoptotic agent linked to a Cre-dependent virus in order to get rid of all the TH-positive neurons on one side. And Maricela Morales tells me we got rid of about 95% of them. Then we included those animals in our experiment, trying to record neurons from them and sorted those cells. And when we do that, what we find is we don't have any impact on the prevalence or any of the measurements that characterize the neurons down here that we would call the non-dopaminergic neurons, or at least the ones that are not classic dopamine neurons, but we almost completely eliminate the cells that we would normally classify as dopamine cells. So about 95% of them seem to have disappeared. So these are sort of classical wide waveform cells, and they are clearly TH positive, I think. So do they show classical prediction errors? The answer to that in this task is definitely yes. So this shows the average firing rate of the reward responsive dopamine neurons at the block where we're going from a small reward, which is a single drop at time 0, to a big reward, which is 1, 2, 3 drops at time 0, half second, and 1 second. And you see a big increase in firing to that second drop, which is really the most unpredictable one, and a smaller increase to the third drop. In each case, the change in firing is maximal initially and then declines. At the same time, in the other well, where we are omitting those second and third drops, you see deep suppression of firing that then comes back up. So you can see it here where I'm just plotting for you the first 20 trials. Here I'm plotting the activity of the same neurons at the beginning versus the end of the block. So big increase to the second drop, a little bit less to the third drop, and then suppression over here initially when we're omitting those drops. And you can also see the same thing here where I'm just plotting the average firing activity in those 500 millisecond windows associated with each drop of reward. And you can see the changes really only happen to the second drop of reward and the third drop of reward. So we have these increases that go down and then suppression that comes back up. The suppression is obviously a lot weaker than the increases, but the two are correlated. And you can see that here where I'm just plotting for you the difference in firing early versus late to unexpected reward versus reward omission. Our signal is not as clean and as beautiful as the iconic example I started with, but the neurons are clearly representing both of these things. So you don't have omission neurons and positive error neurons. In fact, the disappearance of the prediction error here, the positive error, correlates with the appearance back to the cue. Right? So these neurons are showing all the features you would expect for signaling of this classic cache value error. What do they do when we hold the value the same and simply switch what the flavor is that's being delivered? The theory suggests they shouldn't care at all about this, right? Because the animals don't care. They don't show any difference in value when we switch the reward, at least by their behavior. When we look at the neural activity, we find that there is a change. So this is the average firing now in the same dopamine neurons I was just showing you, now at the point where we're making a transition between the different identities of a reward, right? So the size stays the same, but now we're going from chocolate to vanilla or vanilla to chocolate, and you see an increase in firing on trial 0, not only to the second and third reward, but now also to that first one. And we see it not only on the side where we have the big reward, but we see exactly the same thing on the side where we have the small rewards. An increase in firing, and if anything, it seems to persist sort of more trials on that side. You can see the difference also here where I'm plotting the firing activity at the end of the previous block versus the beginning of the next one. So again, you see the increases to each of the drops. And you see the increase in firing not only to the first drop of the big reward, but at the same time to the first drop of the small reward. And these things are in fact correlated in the individual cells, right? So it's not that we're, I think this addresses some concerns that we've gotten that there might be differences in value that we're not able to pinpoint, right? So when we change the identity, we're making one reward more valuable, so we see an increase. But if that was true, then we should see a decrease in the other well, right? Because that would then be less valuable than expected. And we don't see that. Individual neurons are showing increases to both in real time. Yep. So you mean the way the blocks switch? Yeah. So they, every day they started with a different arrangement at the outset, and then we would go from there. So the sequence was generally the same, but one day it might be big vanilla, small chocolate, small vanilla, big chocolate. I mean, there are basically 4 combinations. And the switches were totally unsignaled, and they usually would happen between 60 and 80 trials, but sometimes they were earlier than that, sometimes they'd be later, so it was unpredictable for the animals. Yeah. All right. And then the last point to make is that what we would call the identity prediction errors are correlated with the classical value prediction errors. So it's not that there's a separate population of cells, at least to the best of our resolution to see this. Maybe if we recorded thousands of these things or did an ensemble analysis, we would see that. But for the 50 or so cells we had, those two things are in fact related. So as your identity error is bigger, your error to the unexpected delivery of reward becomes bigger. And importantly, we also see a correlation now inverse with the negative prediction errors. So the bigger the response to an unexpected identity, the bigger the suppression on omission of the expected reward. And this is important, I think, because if the increase in firing on change in identity was salience or surprise or an unsigned error, right, then this relationship should be positive, right? Because omission of an expected reward is a highly salient, surprising event, right? So it suggests that what these neurons are doing when I'm shifting the identity of the reward is actually fundamentally the same as what they're doing when we're adding new rewards or leaving them out. So the neurons are signaling the error in the prediction about the reward, and it's not limited to that error and how it impacts its value. At least that would be the argument. Whether you believe that or not, it clearly goes beyond what is easily accounted for by this model, I think in both directions. So I like this because it seems parsimonious to me that I have this teaching system and it's able to do a lot more in terms of helping me to encode the relationships in the world around us that I know that we are actually learning about. So that's it. Sorry, I went a bit over. I need to thank two people. Mel Sharp, who's a postdoc in the lab, who did the optogenetics work I started with. So she's done a fantastic job and is actually on the job market now. And then Yuji Takahashi, who's a staff scientist in the lab, who's been with me a long time. He spearheads all of the dopamine recording work and did the second study. He's not actually in the picture, but and then also I need to thank NIDA for all the support they give me. Thank you. Almost nobody left early. It's really impressive. You're hanging in for the long run. Sam.
I'm actually not going to maybe counter-argument. I find this data compelling. I think it's probably, as I'm sure you'd agree, it's important not to throw out the baby with the bathwater. So, in particular, it's important to distinguish between the concept of caching, something that's getting cached that's being updated by temporal difference, prediction error, and the nature of the thing that's getting cached. So is it possible to retain the fundamental idea of a TD error, but have it operate over different representations that are getting cached, right? And I think that's kind of the minimal adjustment to the TD error idea. And in fact, that is in fact this idea goes back a long way. So it goes back several decades to an idea that Peter Dan had before he even wrote about dopamine, which is this notion of the successor representation, which is essentially a generalization of the ideas that motivate TD learning, but to arbitrary features. So you're learning some predictive map of features or states in your environment, and importantly, that predictive map or those predictive feature representations can be updated using a form of TD learning, but now operating over the feature space, not over value. So I don't know if that resonates with your...
Yeah, I mean, the problem here is I'm not a trained computational neuroscientist, right? So I realize I am probably testing variants of these models that are now no longer the ones that you guys are thinking about, right? But these are the ones that are, I think, deeply influencing neuroscience, right? So my hope would be that people like you would now help me understand how this makes any sense, right? Because I know it sort of doesn't. And when I talk to Nathaniel about it, he points out, I mean, so one of the problems that has been suggested to me is that if you just have these teaching signals, how do you know what to learn about, right? Like the value prediction error tells you what you're learning about. You're learning about value, right? It's part of its value. So if you're getting these signals in a way that's not bounded by that, then how does a set of neurons know, what to acquire representations for?
The answer to that, which is that the idea of success representation is motivated by the is actually kind of a really interesting mathematical observation that you could represent value as a linear combination of predictive features. So that implies that
that's interesting.
You learn these predictive features, and these features can be basically everything, right? And in fact, it subsumes the original value coding idea, because if reward is one of your features, then what it's going to learn is value, right? But as long as you can learn all these things, just this kind of vector valued prediction error signal that's updating all these different things, and then there's some other area that has to use that information to predict, actually approximate value, and that is itself kind of simple computation. Anyway, we can talk about it more later.
Yeah.
So, yeah, the second experiment is very interesting, but also maybe for some questions. One is when something is associated with punishment, then dopamine neurons tend to get inhibited. So that's also a sensory prediction error.
Sure. Yep.
But it's inhibited.
Yeah.
So,
Right.
I thought it depends which dopamine neurons are recorded.
So the question is, it's fine to have more complicated teaching signals, but if you look at reward or the quality of reward and punishment, all is very messy in that sense. So do you have some simple mechanism biologically how this kind of teaching signal could be?
Yeah, I don't know if it would I don't know if it would qualify as a mechanism. I would have two responses to that. I mean, it seems to me that there's a lot of variability in what people have found in terms of how these neurons respond to punishment, right? And distinguishing that from what they do to rewards and also what they do to salient events, right? This is like could be a cottage industry. So it's possible, I mean, I said very carefully at the end that at the level of our ability to resolve these things, we're not seeing different populations. But maybe if we had more neurons, we'd begin to see separable populations. So perhaps there are different populations doing these different things, right? Alternatively, I would suggest that the dopamine system is really the only place in the brain where we're still trying to figure out functions based on how individual neurons do some giant thing, right? So, you know, we're looking for cells and looking at how one neuron does a set of things and trying to figure out what does the whole area do, right? What if instead we could record 100 of these neurons at once using calcium imaging, right? And look at the ensemble representation, right? So don't you think that maybe there might be a richer form of an error signal in the pattern firing across a group of these neurons that would easily resolve all of these apparent contradictions? Does that make any sense?
Well, I guess that was question in theory.
Well, that's right. But yeah, so, I mean, again, the problem for me is that we're trying to look at these things one neuron at a time. And then we want to argue about how one neuron informs us about the function of the whole population, where, you know, imagine if we had tried to figure out hippocampus, right, you know, without ensemble recordings. You know, we wouldn't be in the same place. So I think there's this, trying to get beyond this model, I think, is what we need to do.
But then going up that, I guess it would be interesting. Those increased response with quality switching might respond positively to punishment.
Yeah. So I guess if you. Right. So the way I see it is that in our experiment, the only place that there's actually a negative sensory experience, like the actual loss of something, is when you don't give the reward, right? So that's unique, not only because it's less valuable than expected and maybe punishing, but it's also unique because it's like a negative. It's a mirror image of a real sensory event. And so that is when you see the suppression and firing. So one possibility is that the dopamine neurons are signaling suppression in the situation where there is the absence of information, right? And so we're actually, one thing that people have asked us to do, which is difficult for reasons I probably shouldn't waste your time with, is why don't you just record during sensory preconditioning, right? Like in the actual first phase. And so we haven't done that, but we're trying to now. And one prediction would be that if you had two cues the animal predicted were associated and you left the second one out, maybe you'd see a suppression, right? My reluctance to do this is I, again, don't think this has to be a single unit correlate, right? It could be a pattern of firing across handfuls of neurons or something. Maybe the reason the value prediction error is so striking and amazing and is present in a single cell, I mean, think of all the criteria that have to line up for one cell to show this entire fingerprint of activity, right? Maybe the reason we see it so strongly in these individual neurons is because it lines up everything that the whole group of them is trying to do, right? Value of rewards to a starving animal is, really gripping. And maybe that's why this has been, such a powerful correlate. Okay, thanks.